Good morning, everyone. Today, I will talk about enzyme biotechnology, about the different methods for enzyme immobilization and its applications. So, what is enzyme? Basically, enzymes are proteins that act as biological catalysts to accelerate chemical reactions. Enzymes are not consumed in chemical reactions. The molecules upon which enzymes act are known as substrate, and the enzyme converts the substrate into different molecules which are known as products. Traditionally, enzymes in free solution react with substrate to result in products. Such use of enzyme is wasteful and particularly for industrial purpose. Since enzymes are not stable and they cannot be recovered for reuse. So there was the need of immobilization. Now immobilization of enzymes refer to the technique of confining, anchoring the enzymes in or on an inert support for their stability and functional reuse. By employing this technique, enzymes are made more efficient and cost effective for their industrial use. Immobilized enzymes retain their structural conformation necessary for catalysis. Now, need of immobilized enzymes, protection of enzyme from degradation and deactivation, reuse of enzymes for many reaction cycles, lowering the total production requirement of the enzyme, low cost of enzyme mediated reactions, ability to stop the reaction rapidly by removing the enzymes from the reaction solution, enhanced stability, easy separation of the enzymes from the products. Product is non-contaminated with the enzyme. Characteristic of immobilization of enzymes. Safe homing means can be separated from the reaction mixture. Reusability, that is, it can be reused again and again. Now, talking about the advantages of enzyme immobilization, stable and more efficient in function. These can be reused again and again. Products are enzyme free, ideal for multi-enzyme reactions. Control of enzyme function is easy, suitable for industrial and medical use. Along with these advantages, it is also associated with some disadvantages like the possibility of loss of biological activity of an enzyme during immobilization or while it is in use. Immobilization is an expensive affair, often requiring sophisticated equipment. Changes in properties, selectivity, mass transfer limitations, problems with cofactor and regeneration, problems with multi-enzyme systems. Now, talking about the classification of immobilization techniques. So, there are various methods to classify the techniques of enzyme immobilization. So, methods for immobilization. Methods for soluble enzyme, it is divided into two types initially: methods for soluble enzymes and method for insoluble enzymes. In case of method for soluble enzymes, two types are there: entrapment and binding. In case of entrapment, there is gel entrapment, fiber entrapment, and microcapsulation. In case of binding, there is carrier binding, cross-linking. In case of carrier binding, four types are there: physical adsorption, ionic binding, chelation or metal binding, covalent binding. And in case of cross-linking, now another type of classification is like it is divided into two types: physical and chemical. In case of physical one, adsorption, entrapment and encapsulation is there. And in case of chemical, covalent bonding is there. And covalent bonding is further divided into two: support and cross-linking. So this was all about the classification of enzyme immobilization. Now you can see here in this diagram, this is the another type of classification. So basically all these classifications are based on these three four points like covalent bonding and entrapment, but the way is different. Like in this type of classification, it is divided into two: irreversible and reversible. So in irreversible, covalent bonding, entrapment and cross-linking is there. In case of covalent bonding, cyanogen bromide activation, peptide bond formation, diazotation, activation by bipolyfunctional agents. In case of entrapment, gel technology, microencapsulation is there. Now the another one is reversible. It contains absorption like static process, dynamic process and reactor loading. So it contains three types like support material, enzyme and surface modifier. Now talking about the applications of enzyme immobilization, there are several applications of enzymes immobilization in different fields like in industrial application, analytical application and therapeutic application. So let's talk about industrial application. Immobilized penicillin acrylase may be used for conversion of penicillin G to 6-APA, a basic nucleus required to produce various semi-synthetic penicillin such as ampicillin, amoxicillin, etc. Production of high fructose rich syrup. Fructose is sweeter than glucose, so glucose is converted to fructose in various syrups for palatability by the use of immobilized enzyme glucose isomerase. Another is use of yeast in the baking and brewing industries because they contain the enzyme for alcoholic fermentation, metabolize hexose sugars to produce pyruvate. But whereas animals convert this to lactate under anaerobic conditions. The anaerobic end product in yeast is ethanol with carbon dioxide being evolved. The clarification of cedar wine's fruit juice is usually achieved by treatment with fungus pectinase. Pectinase area Pectinases are a group of enzymes including polygalacturonase which break the main chain of pectins and pectin esterase which hydrolyze methyl ester. Their reaction releases the trapped particle and allows them to flocculate. Now, another is cheese production. Then, paprine is sometimes used as meat tenderizer. Another industrial applications are washing powder, bacterial protease. So, washing powders incorporated bacterial protease. contaminate in granules which rupture only one contact with water. The enzymes subterminates from Bacillus subtilis mutant and are stable to alkali at high temperature, that is at 65 degrees Celsius. They will attack blood and other protein stains. Bacterial proteases are also used in the leather and textile industries to loosen hair and enable it to be separate from hide. Now talking about analytical application, enzyme, electrode and biosensors. ELISA, basically ELISA is also used. It is a plate-based assay technique designed for detecting and quantifying soluble substances such as peptide, protein, antibodies, hormones, etc. Immobilized heptanes as immunopurifiers, affinity chromatography and purifications. One species may have affinity for the material to be removed from solution purification. Another is immobilized avidine biotin based system in which avidine, that is an egg protein, have avidity for biotin, that is cell vitamin. Biotin may bind with antibodies, proteins, whereas avidine may carry support. Thus, biotinated antibodies may be used to separate specific type of protein from the system. Another application is therapeutic application. In this, the preparation of immobilized enzyme, for example, fibrinolysis, streptokinase, urokinesis in microgranules of cephadics can be used for the treatment of thrombus. Treatment of phenylketonuria. It is the mental retardation state where deficiency of enzyme leads to hindrance of conversion of phenylalanine to tyrosine. The immobilized PAH, that is phenylaniline hydrolase, may be used for the treatment of this disease. Another application is triggered drug delivery. In this system contains the active agent placed subdermally or in other appropriate body sites where it remains passive until a specific molecule appears in tissue surrounding the device. For example, there is a narcotic antagonist known as naltrexone. Another application is artificial organs. The specific enzyme may be immobilized and implanted in the body, may work as artificial organs. For example, artificial pancreas may immobilized be isolate cells of pancreas to release insulin. Artificial liver may immobilize several metabolic enzymes. Artificial kidney may be used as blood purifier by used several adsorbent. So this was all about the the immobilization, enzyme immobilization and its various applications in different studies. Hope you understood. Thank you.